Hello students and welcome back to the interesting world of toxicology. Today in this video we're going to talk about the mutagenesis, the carcinogenesis, the teratogenesis, as well as antagonists and antidotes to get rid of those toxins and the risk assessment. So let's begin. Hello to you all, I am Tommy. I am a human nutrition student and researcher at the university. What I do in this type of videos is to synthesize what I learn at the university so then I can explain and teach you the best and most valuable information you need to know. So let's begin with this video. Mutagenesis. As an introduction, it is a genetic modification that is transmitted to the next generation. The xenobiotics can induce mutations indeed. And here we encounter the adducts. These are compounds that form a covalent bond with DNA molecules. The adducts are like salt in a zip of a beach sweatshirt. It binds to the guanine and it leaves the double helix open, so it's difficult to close properly. These are not mutations per se. They are lesions that may be pre-mutagenic. So adducts, pre-mutagenic. Types of mutagenic effect. We have genetic mutations and chromosomal mutations. Genetic mutations are small in size. They affect only a few DNA pairs. And here we have the substitutions and the mismatches. Substitutions can be either by transition, which is a purine or a pyrimidine replaced by the same kind, so purine for purine and pyrimidine for pyrimidine, or a transversion, which is a purine replaced by a pyrimidine or vice versa. Subtypes of substitutions can be silent, when there is a change in the codon that creates an amino acid, but there is no change in the amino acid per se. Erroneous, when there is a change in the codon and there is also a change in the amino acid represented by that codon. The codon is a triplet of nucleotides. And the random one, which is the triplet that becomes a terminator codon. So it ends the protein synthesis and it creates a shorter than normal polypeptide, so not a full protein. Mismatches, on the other hand, they are the loss or addition of several bases. There are changes in the reading pattern of the DNA transcription, and these can be reversible by back mutation. Then we have the chromosomal mutations. They affect a large fragment of DNA. Indeed, chromosomes are very large parts of DNA. Numerical or structural. The numerical ones are loss or gain of one or more chromosomes. And there are failures in cell division here. Down syndrome, for example. Structural. There is a rupture of two chromatids, and this can create chromosomic mutations, which are changes in several genes, or chromatistic mutations. These are changes in a particular gene. What's the impact on human health of the mutations? In somatic cells, they can create neoplasia, so tumors, alterations in embryonic development, and affecting germ cells, so sperm cells and egg cells. They can be transmissible to offspring or not. The mutagenicity assessment, there are various categories of tests that detect the gene mutation, that detect the chromosomal alteration, or that detect an interaction with the DNA. Now talking about carcinogenesis, as an introduction, chemical carcinogenesis, we have to understand the carcinogenic agent. So it is a chemical structure capable of causing a neoplasm, a tumor. And carcinogenicity is not an intrinsic property of a chemical compound. So it's not because of one molecule only. There are various factors. And a human carcinogen is a molecule that needs an evidence to be determined as a human carcinogen. There are three types of evidence: sufficient, in which there is a causal relationship between the agents and human cancer; limited, where there is a credible causal interpretation, but there are also alternative explanations that may be possible; and inadequate, there is little relevant data available. Mechanisms and stages. So we have three: initiation, from a normal cell to an initiated cell; promotion, so the creation of a benign tumor; and the propagation, where it goes to a malignant tumor, so proliferation of the tumor. There is an exposure to carcinogens, then a metabolization of the agent, and after that, there is an interaction between the agent and the cell components, so the DNA. This will lead to growth of abnormal clones from initiated cells here, and growth of the tumor and spread to all parts of the body. So as you can see, there are some molecules that act as carcinogens. What are the types of carcinogens? By action type, they can be initiating, so in the initiation stage here, from normal cells to initiated, promoting agents, so they promote the growth, or complete carcinogen. They induce the cancer from normal cells, so initiating and promoting. Carcinogens can also be classified if they are genotoxic or not. Genotoxic carcinogens behave as initiating agents. They interact with the DNA and chromosomes, creating neoplasms. They can be organic, such as polycyclic aromatic hydrocarbons, aromatic amines, drugs, nitrosamines, and mycotoxins. And this can be found in the food processing. Or inorganic, such as cadmium, chromium, arsenic, or nickel. Non-genotoxic carcinogens, they don't react with the DNA, but they can also induce tumors. Indeed, there is a cytotoxicity and proliferation, a chronic inflammation, and hormonal influences. These are mainly found in the environment, such as dioxins, phthalates, DDT, phenobarbital, and tetrachlorodibenzophosphorodioxin. Assessment of carcinogenicity. 3 methods here. We have the in vitro cell transformation assays, in vitro carcinogenesis assays, and epidemiological studies, which is the only tool we have for human studies indeed, because we are not going to induce a tumor on a human being. And then teratogenesis. So it's a process that creates malformations, not only physical, but also lowering the IQ, for example. Agents that alter the normal development of the embryo. These are teratogens. The principles of teratogenicity is the embryo's genotype plus the environmental factors that are all around the individual, and also the idiosyncrasy of the individual. Affected development stage will differ because it's not the same the beginning versus the end of the pregnancy. Teratogens will use specific mechanisms to attack cells and tissues. So what are the factors modulating teratogenicity? Species, susceptibility, critical periods in gastrulation and organogenesis. Dose dependence, remember Paracelsus. There are no acceptable daily doses for these compounds, though, same as carcinogens. Teratogenic mechanisms, mutations, chromosomal breaks, alterations in cell division, osmotic imbalance, and enzyme inhibition. These are all mechanisms that can lead to a malformation. There is also the influence of maternal and paternal toxicity. So how healthy is the mother? There are maybe repercussions not only for the fetus but also for the mother. And taking a look at the male sex, how healthy is the father? And there is a worldwide decline in sperm count and sperm quality. So this is a factor we need to take in account. Manifestation in developmental disorders. We have malformations, growth retardation, embryonic lethality, and functional impairment. So the malformation can be the majority spontaneous, but also non-spontaneous. And these can induce the apoptosis, which is the programmed cell death. And if a cell in an organ dies, that organ is no longer developed. Growth retardation, the fetal size, it's very important, this parameter for the assessment of a teratogen. Many teratogens retard the growth. So if the fetus is small, probably there is a teratogen acting. Embryonic lethality. In the early days, the embryo is reabsorbed, so nothing really happens. Before 20 weeks, there is a miscarriage, and in advanced stages, there is a dead fetus. Functional impairment, non-structural alterations, such as motor skills, learning, emotion, and behavior. As we said, also a lower IQ, possibly. Assessment of teratogenicity. We have 3 methods here, so the animal testing, the in vitro methods, using organ or embryos, cell cultures, or subvertebrate species, and the epidemiological studies that can be done in humans as well. The main teratogenic xenobiotics are this one, including cocaine and ethanol, yes, alcohol. Now let's talk about antitoxic therapeutics. So, as an introduction, we have to resolve life-threatening situations. By neutralizing or preventing the toxic effect, we cannot let the effect happen. So there are some principles of the antitoxic process, in which the intensity of the toxicity is related to the dose. Guess what? Paracelsus again. And the effect depends on the route of administration. It's not the same inhaled than oral. And the intensity is determined by the relation between time and distribution, and also by the permanence of the xenobiotic in the body. So here we have two solutions: to use antagonists that will act on the receptor, or antidotes that will act on the toxic agent. Antagonists will be specific, they will compete with a xenobiotic via blocking the receptor or displacing the toxicant if it's in the receptor already. The substance in higher quantities always wins the competition, so make sure to use a lot of antagonists. But antagonists can also be non-specific, and this is to stimulate a counteracting organic activity to either overcome or even override the functional defect. This is, for example, when we use the adrenaline for anaphylactic shock. The anaphylactic shock tends to lower the heart rate, tends to bronchoconstrict, and to vasodilate. So with the adrenaline, what we do is the reverse. We vasoconstrict, we bronchodilate, and we increase the heart rate. Antidotes, they act directly on the chemical structure of the toxicant, and there is an inactivation or prevention of the toxic action. Indeed, we can do this with the destruction of the toxic structure, neutralization of acids using bases, and of bases using acids, or maybe using oxidation. For example, potassium permanganate is used for alkaloids and cyanides. Then we can also block the toxicant. Ways of blocking is adsorption, for example, using activated carbon, but this must be done quickly because if it is already absorbed, then it's no longer efficient. Via absorption, using an albinous water for metals, insolubilization as well, to create a non-absorbable precipitate, or chelation for heavy metals. And then we can also create a less toxic structure. For example, sodium thiosulfate will convert cyanide to sulfur cyanide ions, so lowering the toxicity. So what is the therapeutic procedure? Steps to follow. Stabilization first is very, very important to prevent worsening. Then, after that, we can complete the assessment. Tests, severity of clinical effects, and possible traumas and complications. Then we take measures to decrease the absorption. And then we try to improve the toxicant clearance, so the elimination. And we may consider the use of antidotes if there are any available, because only 5% of the cases have an antidote. Fundamentals of antitoxic therapeutics. So the dependence factors of the toxic effect are the concentration of the toxicant at the receptor. This is called the toxicity threshold. And remember, concentration equals dose, so Paracelsus. And time above this toxicity threshold. So for how much time have you been exposed to a toxic concentration, a toxic dose? Reduction of the toxic area can be done by lowering the toxic arrival speed at the receptor, so acting on the absorption, using adsorbents, emetics, laxatives, and chemically acting antivenoms. If it's inhaled, the way of administration, then what we can do is to immediately take out of the area, because it's very, very dangerous. It's a very fast way of absorption. If it's ingested, we can use adsorbents, for example, gastric lavage or activated charcoal, as we said earlier. Emetics, they are generally not recommended, only if the gastric lavage is not possible. The use of laxatives for the fat-soluble toxins, and for this we use the saline-type purgatives, and also antidotes, insoluble compounds, oxidation, precipitation, and combination. Then we can also increase the degradation, the blockage, or the rate of elimination. We can do this by targeting the speed. Increase phase 1 and phase 2 reactions, the destruction of toxic structures using antidotes, and displacement of the toxicant using specific antagonists. To increase the elimination rate, we can prevent the reaction with the receptor. So here we are going to use antagonists and antidotes, either by inactivation or destruction, which is the most effective. And also the displacement once it's already associated with the receptor. And here we use specific antagonists. And again, the one with the highest concentration wins. And then another thing we can do is to raise the toxicity threshold, so that at the same concentration there is less toxicity. For the threshold, we use non-specific antagonists. They act on the antagonist system by competing with the metabolism of the xenobiotic. And again here, if we use a lot of concentrations, we will get the effect we want. And now let's talk about risk assessment in food toxicology. It is a collection of information on the toxicant To determine the risk, here we have to differentiate between the hazard, which is the ability to cause harm, and the toxic risk, which is the likelihood that a hazard can cause damage or harm. If there is little data, there is uncertainty. And knowing this, we can proceed to the risk analysis. Risk analysis is divided in risk assessment, risk management, and risk communication. The risk assessment is divided in hazard determination, in which there is a determination of the agents that may cause adverse health effects. They can be biological, chemical, or physical, and they may be present in the environment or in the foodstuff. Then there is the hazard characterization, in which we have an assessment of the harmful effects that those hazards, that those agents that we determine can do. It can be qualitative and or quantitative. There is a data collection, so we have to ask ourselves these two questions. Does it produce harmful effects? Okay, how do you become exposed to that substance then? And this is used to characterize and assess the hazard to calculate the dose response in the population. So the dose at which the majority of people of a population will get the effect. And this data is allowing us to assess the toxic effects, to decide on labeling, restrictions on use, and also the permitted limits of a substance in food. And then there is the exposure assessment following this question. Assessment of the likely intake of biological, chemical, and physical agents. It can be quantitative or qualitative, and there is an exposure assessment strategy for this, which is to calculate the toxic agent based on the population diet. And it's always better to overestimate, so to take in account the hypersensitive. Remember the Gauss bell. So based on the type of substance, we have biotoxins. The steps to follow are to know the normal contents in the food, to estimate the maximum daily intake for the population, to estimate the intake in high consumption situations as well. Regarding food additives, there are a lot right now. There is an excess of them, and many of those are not needed. But if we remove some of those, such as nitrates, there can be very dangerous consequences. Then with pesticides residues in food. There is a problem here with farmers, because they misuse pesticides. We cannot use the same pesticides for all foods, because some foods may reach toxic doses of those pesticides because of different sensitivity of each food. And then environmental pollutant. Here we follow the One Health concept, and we talk about phytosanitary residues. There can be local or regional emissions. Sources of contamination may differ. Annual production and the life cycle of the substance is understood here. There is a monitoring and prediction to assess the human exposure and also assess the toxic effects, so to calculate the dose response and the NOAEL, which is something we are going to view it right now. In the risk characterization, which is the estimation of the likelihood of a hazard occurring and at what quantities. So here we have it, the no observable adverse effect level, NOAEL, and it is used for the hazard assessment. NOAEL must be divided by an uncertainty coefficient. So what will we do? We take the coefficient between the interspecies jump, which is 10, and the intraspecies jump, so from animal to human and between humans, and it is 100, 10 times 10. So knowing that, the acceptable daily intake, ADI, is the NOAEL divided by 100, and it is defined as the maximum daily intake of a substance that can be consumed without causing harm over a lifetime. Very important. The ADI is for food additives. For the other contaminants, we use the TDI, which is the tolerable daily intake. There is no NOAEL or TDI for carcinogenic substances, only recommendations, because it's unacceptable to have carcinogenic substances in our food. And then we have the margin of safety, which is the estimated daily intake versus the ADI or the TDI. So estimated daily intake divided by TDI. Tolerable daily intake. If this value of the margin of safety is less than 1, there is no risk, the intake is less than tolerated, and if it is higher than 1, there is a risk because the intake is higher than the recommended. And then we have the risk management, consideration of policy options following the risk assessment. So after doing this, we consider what to do, including the implementation of regulatory control measures. And here there is the thing of emerging risk, according to the EFSA, the European Food and Safety Agency. New hazard of significant exposure, new or increased exposure of a known hazard, and new or increased susceptibility to unknown hazard are all emerging risks. And last but not least, we have the risk communication. It's very, very important. It is an interactive exchange of information and opinions on risks, and it involves risk assessors and risk managers, but also consumers and other interested parties. Okay, that's it for this video. I hope you understood everything I said. If you didn't, there is a comment section down below. Don't forget to like, subscribe to the channel, and share this video to your friends and family to spread its knowledge to them as well. Thank you so much for watching, and I will see you in the next one. Ciao!